The first step in epinephrine signaling occurs when the hormone binds to an epinephrine receptor on the cell surface. The hormone triggers the receptor to change shape, converting the receptor to its active form. The activated receptor triggers a cascade of events within the cell, beginning with the activation of a G protein. The G protein binds to the activated receptor, releases GDP, and takes up a molecule of GTP. After taking up GTP, the G protein is released from the receptor and splits into two parts. One of the parts is activated and continues the signaling cascade. Shortly thereafter, the hormone leaves the receptor and the receptor reverts to its inactive form. The G protein activates an enzyme called adenylyl cyclase. When activated, adenylyl cyclase converts a large number of ATP molecules into signaling molecules called cyclic AMP. Because cyclic AMP carries the message of the first messenger, epinephrine, into the cell, it is referred to as a second messenger. In response to an internal timer, the G protein soon inactivates itself by cleaving GTP, and the subunits reassociate. With the G protein no longer attached, the adenylyl cyclase deactivates and can no longer convert ATP into cyclic AMP. The cyclic AMP molecules produced by adenylyl cyclase continue the signaling cascade by binding to an enzyme called protein kinase A. This binding causes protein kinase A to separate into subunits, two of which are catalytically active. The activated protein kinase A subunits perform chemical reactions in which they add phosphate groups to another type of enzyme called phosphorylase kinase. The addition of the phosphate groups activates phosphorylase kinase. The phosphorylase kinase phosphorylates another enzyme in the cascade called glycogen phosphorylase. When phosphorylated, glycogen phosphorylase also becomes activated. In its activated state, glycogen phosphorylase produces the cellular response to epinephrine, breaking down glycogen into its component glucose molecules. During the process, the enzyme adds a phosphate group to each of the glucose subunits. Another enzyme, not shown, removes the phosphate groups from the glucose molecules. Without the phosphate groups, glucose molecules can be transported across the plasma membrane of the cell. Once outside of the cell, the glucose enters the bloodstream and is taken up by other cells and used as a fuel. This fuel represents a key component of the epinephrine-induced fight or flight response.